All right, everyone, can you hear me okay? Anyone can just unmute yourself and let me know. And if you're not uh, like muted, just keep yourself muted. Otherwise, there can be echo can happen.
You're
Yeah, we
good.
can hear you.
You.
All right, great. We'll wait for a couple more minutes. Maybe some other folks will join. So let's just wait for 1 or 2 more minutes and then we'll get started. How is it going, Joe? How was the snow day, though? Sudden snow day. Are you all safe, warm?
Warm as long as we're not going outside.
I know, exactly. All right, I'm going to share my screen. If you cannot see the screen, you should see the slides. And if you cannot see the screen, then let me know. All right. All right. You should be able to see the screen and then the PowerPoint, actually, the blood PowerPoint. All right, so. Okay, so we'll get started, and as people will join, they can hear us, and also this will be a recorded one, so you can hear me. You can hear this later also. So this is an optional class. I could have just given you a snow day, but as you know, these classes have so much content. So if we can, I mean, it will be good now, but then later on, everything will accumulate together, and that will be painful for you people. So what we discussed on Wednesday was blood. We started blood. We spoke about the functions of blood. We spoke about the formed elements of blood. Someone remind me or remind all of us, what are the three formed elements of blood? Anyone can unmute and just let me know. You keep yourself muted, otherwise things will echo. Whoever is answering, just unmute yourself. The three formed elements of blood.
Erythrocytes, leukocytes, and platelets.
Absolutely. Erythrocytes or RBC, red blood cells, leukocytes or WBCs or white blood cells, and then finally the platelets. Why they are formed? Because they do have a certain form, shape. And erythrocytes are approximately 45%. You have done this experiment in lab, hematocrit. And on top of that, there will be this small buffy coat, which is the leukocyte and platelets, which is less than 1% of blood. And then on top of this clear fluid, which is the plasma. So we did talk about characteristics of plasma, what are the composition of plasma, and then last day we spoke about RBC, erythrocytes. Specifically, important thing is their shape, the value of, like, because they carry oxygen and they carry that with the help of hemoglobin. So what is the value that an adult person should have? How is the structure of hemoglobin molecule? All of this is important. And then function of erythrocytes, how are erythrocytes produced? All red blood cells are produced from hematopoiesis, but then the production of erythrocytes is called as erythropoiesis. Then we'll get into, this is so big. Anyway, and then the erythropoietic mechanism with the help of erythropoietin hormone. Where is this hormone secreted from? Erythropoietin, this particular hormone?
The kidneys.
Kidneys, awesome. So erythropoiesis is secreted from kidneys. And then We spoke about the regulation of this process, how erythrocyte destroyed, and then the disorders, mainly anemia. Your case study is actually based on anemia. And then now we did this concept checks, and now today we will be talking about leukocytes or the white blood cells, right? So we'll talk about the structural characteristic, functions, how are leukocytes produced, the disorders, and then finally platelets. So leukocytes or WBCs are the only formed element, and these are actually truly cell because they have nuclei and organelles. They make up approximately less than 1% of blood, which is 48,000 to 10,800 WBC per microliter of blood. Remember, microliter means it is even 1,000 part of an ml, so it's a really tiny amount. This number is super critical. You should remember this number, what the range of normal WBC. What WBC do? Like, what's the function? It helps us to protect against diseases. Right? So leukocytosis is when WBC count goes over 11,000 per microliter. So it crosses this normal level. Then we call that condition as leukocytosis. Well, this is not so much an abnormal condition because leukocytosis can increase with response to infection. Like even if we have any form of infection, leukocytes actually can increase. Two categories of leukocytes: granulocytes, where the cytoplasmic granules are visible. Three subtypes of granulocyte: neutrophil, eosinophil, basophil, and then a granulocyte, which does not contain cytoplasmic granules, lymphocyte, monocytes. Use this mnemonics also in lab. You can use this: never let monkeys eat banana. N for neutrophil, L for lymphocyte, M for monocyte, E for eosinophil, B for basophil. The 5 types, and these are the two major types: granulocyte or agranulocyte. So if I put this in a differential count, what I will see about in terms of this number, 50 to 70% of WBC in our body is basically neutrophil. You should remember this particular percentage. And then eosinophil, and then basophil. You don't have to remember specifically this one, these ones, but the next most abundant one is lymphocyte, which is 25 to 45%. You should remember this as well. So the two most abundant ones, the most abundant granulocyte is neutrophil, most abundant agranulocyte is lymphocytes. Neutrophil approximately 50 to 70, lymphocyte 25 to 45%, and then finally monocytes. Okay, now they have their separate functions and their different kinds of structure also. So we need to know that, remember that. Firstly, granulocytes, 3 types: neutrophil, eosinophil, basophil. They are larger, shorter lived than RBC. They are lobed, and cytoplasmic. They have these granules that can be stained. All are phagocytic to some degree. Phagocytic, this particular term, this means that these leukocytes are capable of digesting any foreign infection. They sort of, suppose this is a WBC and they see a foreign, maybe bacteria or microbe something, they can really form stuffs like pseudopods and then eat up this whole microbe, right? So that's what it is called as phagocytosis or phagocytic called. So they can really digest, because I will just talk in a second, like I'll show you in a second, they do have certain kinds of enzymes that is capable of destroying this kind of foreign infections. First, the most abundant one, neutrophil, 50 to 70%, twice the size of RBC. So it's bigger than RBC. It has these granules, but most importantly, the enzymes that I was talking to you about is called as defensin. Okay, this is a hydrolytic enzyme or antibacterial or antimicrobial enzyme. Okay, so this defensin molecules, whenever we have some form of infection, neutrophil is the first one that will come and try to attack that situation or control that situation. What it will, suppose you have a bacteria in your body, the neutrophil will be there first, and then will start secreting defensin so that that bacteria can be phagocytosized. If not, if they cannot do it, then only they will go for other levels or other types of defense mechanisms. The way you will identify them, because these guys are polymorphonuclear leukocytes, PMNs, or polys means they have multiple nuclei lobes. It's one nuclei, but it is divided into approximately 3 to 6 lobes. You've seen these slides and models in lab already. Eosinophil is the next one, 2 to 4%. This guy has two lobes, not three. They specifically have two lobes, looks like this earmuffs, right? That covers our ear. Now, these guys also have digestive enzymes, but they are specifically targeted to large parasitic worms. Any parasitic worm enter our body, then a eosinophil will be there and will try to digest it with these digestive enzymes. They also have some role in allergy and asthma. So neutrophil, eosinophil. Third is basophil. This is an interesting one. This is the rarest one, really less in number, half to 1% of leukocytes. Their nucleus is deep purple with some constrictions, but sometimes their nucleus are not even visible properly. They lack granules, and the most interesting thing they have is called as histamine. Okay. What is this histamine? Histamine is an inflammatory chemical that acts as vasodilator and attracts WBCs. Means, suppose you have an infection somewhere. So you need to have more blood in that area. Why? Because more blood means you will have more WBC in that area. So you need to take care of that infection. So histamine actually will work. It will work like an inflammatory chemical. It will inflammate the vessels, the blood vessels, so that more blood can come into that area and they take care of that infection. So more WBCs are available in that area. Another thing I would like to mention, they also secrete a substance called heparin. Okay, write this down. Heparin is a molecule that is a natural blood thinning substance. So if our blood for some reason gets really thick, viscous, then basophil starts creating this molecule called heparin. This is actually a natural blood thinning agent. If a person has, suppose, heart blockage in the heart arteries, then doctors typically prescribe them heart thinning medicines. But we do have something like that naturally present in our body. So when heparin actually stops responding properly, then only we have to be given this kind of thinning medicines from outside so that blood do not clot or do not form clogs within the arteries, particularly heart arteries. And they're functionally very similar to mast cells. We'll know about that in the immune system. So these are the granulocytes. Next are agranulocytes, lack visible cytoplasmic granule, two types mainly, lympho and monocyte, right? So first one is lymphocyte. This is the second most abundant WBC, accounts for 25%. The way you can identify them is pretty easy. They will have this huge, big lobe of nuclei, okay? These guys are typically found because they are also called as lymphocytes. So they are typically found in the lymphoid tissue, for example, spleen, lymph nodes, which is part of our immune system, but they're also seen in blood as well. Lymphocytes are two kinds, T and B lymphocyte. T lymphocytes are the most specialized WBCs. They act against specific virus infected and tumor cells, that they're so specialized that their job actually cannot be done by anything else. T cells are very, very specialized in their immune response, right? B cells are also specialized, but they create a stuff called antibiotic. Antibiotic also can digest any foreign infection, any antigen, all right? So both of these guys are super specialized in their function. The second one of agranulocyte is monocyte, right? These are big, the largest of all nucleocytes. They have pale cytoplasm. The way you can identify them, they have a U-shaped nuclei, sometimes kidney-shaped also. And these guys work as macrophages, means they are the most active phagocytic cell. Suppose you have a virus, bacteria, parasite, some form of chronic infection that is in your blood. These guys will actually form that pseudopod and will try to digest it. Okay. So monocyte. So these are, you should know all of these subtypes. And I said the major percentages, the big two ones, and their function and their specific. I'm not going to ask you to identify them in lecture, but most importantly, you will be asked about their function and especially what kind of enzymes, what kind of product they produce. For example, basophil produces histamine, heparin, stuffs like that. Eosinophil produces parasitic digesting enzymes. Neutrophil produces defensin. So these type of specific questions will be there. Okay. Next, we'll talk about how are leukocytes produced. So leukocytes are produced by the same process called leukopiosis. What is the process specifically for erythrocyte? What is the
Erythropoiesis.
erythropoiesis? Yes. Poiesis means synthesis. So that was red blood cells. So erythropoiesis, and this is leukopoiesis because this is we are producing now leukocytes. Two pretty important factors in this process, interleukin and colony stimulating factor. Now, production of leukocytes is little bit complicated than erythrocytes. They follow two paths. First path is the one that produces lymphocyte, and the second part, everything else, is produced by everything else. Right? So these lymphocytes are produced from lymphoid stem cells, and everything else is produced from myeloid stem cells. All right. So again, we have this cell called hematocytoblast, which is the mother cell of all the blood cells. Now they get divided into two generations. Okay. First becomes the myeloid stem cell, then becomes the lymphoid stem cell. Now the myeloid stem cell now goes to different series of divisions and development. Finally, they produce eosinophil, basophil, neutrophil, monocyte, all the 4 types of WBC. Monocyte later becomes macrophage when needed. Now, the other line, the second line, this is the line number 1 and this is line number 2, becomes lymphoid stem cell. You should know these two lines, the stem lines. And then that becomes T and B lymphocyte precursor. Finally, it develops into T lymphocyte and B lymphocyte. Now, that is not the final work actually, because when these T lymphocyte or B lymphocytes are active, T lymphocyte actually produces effector T cells. Effector T cells are the ones that actually do the work, not T lymphocyte. Similarly, B lymphocyte, when it has to active, when it had to actually have to work, then it forms plasma cell. This is the cell that produces antibiotic. Okay. So they actually get converted into another active form. So T and B lymphocyte is not the final form. You should know, obviously, this particular mother cell and the two stem lines. You don't have to remember, they don't need to remember these intermediate steps, but the final step, and then for lymphocytes, the final effector or the more active cells actually. Okay, so all these. Next, we'll talk about the disorders. Two kinds of disorders can happen. Either we can produce too much of WBC or can do less. Another thing is called infectious mononucleosis. This is a viral disease. It's not necessarily it can be fatal, but not necessarily. But the other one, leukemia, is basically blood cancer, which means when we are overproducing abnormal amount of WBC. So the WBC line is muted and we are kind of producing some abnormal amount of that abnormal WBC. It can be acute and quickly advancing in children, but for adults, it could be slowly advancing. Okay. Because the proliferation of this case in adults happens in the next stage. So some of these late cell lines are actually go into like cancerous line, or they get muted and then they start overproducing these. So this takes time to get into the later part of the developmental pathway. But in kids, actually, it's pretty acute. So immediately it produces these stuffs. But anyway, so the acute form or the quickly advancing part is seen in kids, but in chronic one, which is a slowly advancing one, is seen in older people. Exactly the opposite type of disease is called as leukopenia, which means we have really low number of WBC. And this can be not necessarily always it is natural. Sometimes if someone is taking too much of anti-cancer drug, then the job of anti-cancer drug is to reduce the number of WBC. Then we might see this condition because of the drug. All right. The second one, last formed element is obviously platelets. Now, platelets are not full cells. They are fragment of this thing called megakaryocyte. They're small and they have several clotting chemicals, including serotonin, calcium, several enzymes, ADP, platelets. Why? Because their function is to form temporary platelet plug. So when you have a tissue injury, I'm sure you have seen that you start losing blood immediately. And after certain few minutes, that blood clots. And why? Because platelets help platelets kind of gathers in that area and makes sure that that area is temporarily repaired, because tissue completely, it will take time for tissue to completely repair. All right. But in normal condition, this is an important thing to remember. In normal condition, you cannot allow platelets to be active. Why? Because if it's active, it will start clotting blood. So, and there are two main chemicals that are involved into making sure that platelets are not active, which is nitric oxide and prostacyclin. They constantly like circulate within our blood vessels, making sure that platelets are not active and only active when they are really needed to be active, actually. All right, so platelets formation is regulated by a hormone called thrombopoietin. And again, it starts from this hematopoietic cell or hematocytoblast. It goes through several developmental pathway and finally develops the stage 4 megakaryocyte. When it reaches here, after this is till here, it's kind of a normal cell. But after this, it fragments into lots of pieces and form platelets. Okay, so from stage 4 megakaryocytes, actually platelets develop. They don't live very long, only for 10 days. And we have approximately 150 to 400,000 platelets per ml of blood. This is the entire summary of what we just spoke about. Pay attention to this table. It's a good, nice table, everything summed up together. Okay, now questions for you. So mainly the questions, like I said, will be dictated to or will be coming from the functions of these WBCs. So the first question, an elevated neutrophil count would be indicative of what among these options?
C.
C and A, both actually will be applicable here. A and C, yes, and as well as A, both can be applicable here. All right. So A. Antihistamines counter the action of which white blood cells? Antihistamines. So a certain white blood cell produces histamine. So antihistamine is going to counter their action. So which one is that?
Basophils.
Basophils. What is the function of histamine? What does it do?
Doesn't it? Go ahead.
I was gonna say, like, that is an inflammatory chemical.
Yes. Anyone you would like to add? I don't know who's the other speaker. Anything you would like to add?
I was gonna say that it was an inflammatory chemical too.
Okay, thanks, Autumn. And so basically, what does it do whenever we have an infection? Okay, suppose you have an infection somewhere, and some bacteria have accumulated. We need antihistamines will try and work and will make our vessels wider. Why? Because that will make sure that more blood is there in that infection site, and more blood means more WBC, so that bacteria can be tackled. All right, third one, leukemia is a general descriptor of which of this following disorder?
J. J.
J?
An overproduction of abnormal leukocytes.
Overproduction of abnormal leukocytes. Yes, it's blood cancer. So when we have abnormal mutated leukocytes overproduced, that's leukemia or blood cancer. Right. Which of these cells is most important for fighting parasitic infection? It's an easy one. Specifically parasites.
Monocytes.
Nope. Eosinophil. What does monocyte do? What does this do? Eosinophil is for parasitic infection. What does monocyte do? It's a big one. Largest in size among all the other WBCs.
They differentiate into macrophages.
Yes. And what does that do?
That fights against like viruses and chronic infections.
Yeah, so basically when we have a virus or bacterial infection, monocytes will become phagocytes or macrophages. So what it will do, it will simply, suppose you have a bacteria and here is a monocyte, it will start forming this stuff called pseudopod. It will take this guy inside and it will digest the entire, basically they have lots of hydrolyzing enzymes, so it will digest the entire bacteria, making sure it's gone from our body. So that's what it means. It's formed several pseudopods. It's something very similar how amoeba works, if you don't know. The next thing is about hemostasis. Hemostasis is about how blood clot is formed. So we'll talk about hemostasis, hemostatic disorder, and then finally about blood groups, and then how to replace blood volume. This should not be very complicated because I know you already have done the lab. And after you do the ABO blood experiment, then ABO blood group is easier to discuss. So hemostasis is basically a fast series of reactions for stoppage of bleeding. Requires a series of clotting factors. 3 steps: vascular plasm, platelet plaque formation. Remember, we just spoke about this, and then coagulation or blood clotting. Okay. Firstly, we have a spasm, we have an injury, smooth muscles will contract, causing vasoconstriction. And spasm will result in this vasoconstriction, and then immediately these platelets will like run into that area, to clog it. Okay. And so the first step is caused by the vascular spasm. In the second step, the platelets will run into that area. Platelets will stick to collagen fibers in that particular area. They immediately will not stick because also prostacyclin and nitric acid has to immediately it allows a little bit time for platelets to become active because the collagen fiber has to be exposed, because collagen fiber is like a like something that seals actually. Okay, it's that the tissue that will basically seal this gap, and platelets will then dump above that. So it allows prostacyclin and nitric acid waits a little bit to act so that immediately platelets are not active, so that the collagen fibers are ready. And when that happens, then the platelets actually will come into that area. There's a special factor called von Willebrand factor that stabilizes this platelet collagen addition, because when the collagen is formed already, then the platelets has to adhere or like sit on top of that properly. Otherwise, the plug will not be formed and we will continue to bleed. Well, there are many factors that works in this. ADP causes the platelets to stick and release their content. There is serotonin, thromboxane 2 enhances the vascular spasm as well as the platelet aggression. You should know these two names, and also ADP. Now, this is a positive feedback cycle. You know this already from 208. As we release, as we have a tissue injury and we start losing blood, platelets will continue to be secret and start will continue to block that area until the blood is stopped secreting, or until that seal is totally done. Then it will continue to secrete more and more platelet plugs. So important thing to remember that platelet plug formation, this whole thing is a positive feedback cycle. Finally, the blood will clot. Coagulation means the reinforces the platelet plug with, there is another molecule now after this collagen and then platelets have like clubbed together, then another protein called fibrin is protein. You remember this name, fibrin is the most important protein in terms of blood coagulation. And then finally, the blood is clotted. When fibrin is there, forms a mesh with the blood cell, the platelets, collagen fibers, everything. So then the clot is formed. Okay, blood now is converted to liquid to gel. There are several number of clotting factors that can actually affect this process, and they are numbered from 1 to 13. This is 1 and this is 13. There are 13 actually that we know so far. The vitamin K is also important in this in this context. All right. Here is the list of blood clotting factors. The first one, fibrin or fibrinogen, that we just spoke about. Then other than that, there is prothrombin, there is tissue factor, calcium is also important. You should know these three names for sure, and some others if you can remember, but these two are the most important: prothrombin, fibrinogen, and calcium. All right. So that's how the clotting will happen. After that, now clot will stabilize and has to be removed because the clot cannot stay there, because underneath the tissue will start repairing. So after that, you will have, I'm sure you have seen that after a few days that the scab that is formed under injury, that falls apart, like that falls down, that's like gone. And that is what it is called as clot retraction. And finally, the fibrinolysis happens. Clot retraction simply means that the serum from the clot is squeezed out. It draws, like ruptures the blood vessels. And because at this time, vessels is already healed, right? So there are certain platelets, derived PGDF or platelet derived growth factor is released by the platelets, stimulates the division of this endothelial cell. Can anyone remind me why I'm talking about endothelial cells here? Tissue 208. Why I'm talking about endothelial cells here? No? Because the blood vessels internally are formed of endothelial cells, smooth muscle endothelial cells. Okay, remember not tissue, we discussed this partly in tissues and mainly in also later than in the muscles chapter, smooth muscles. So internally it is made up of smooth muscle endothelial cells. So therefore, they are the ones, this platelet derived growth factor will start stimulate the division of those cells. Basically, those cells need to grow again so that the tissue is like damaged. So suppose you have a gap here, and then these muscle cells will start growing so that this gap is sealed now, right? So they will start multiplying, basically dividing, like will start dividing, and then that will restore the blood vessel back into its original position. Now, fibrinolysis is important here. Why? Because when the clot is done, like the repair is completely done, then fibrin will not stay in that place anymore. So then they have to be lysed, right? So process by the clots are removed. So this is why when fibrinolysis is done, then that clot will fall apart from the tissue, like damaged area. Begins within 2 days, but it will stay there and it will not happen completely. Fibrinolysis will not happen completely until the entire clot is like repaired, all the tissues are repaired and everything is done. Then the clot, the scab will fall apart. All right, so I will actually continue from here on Monday. Now remember, Monday, I do have a medical day, and I did say that in my announcement before. So I will be posting a recorded lecture, which might actually repeat some of the stuffs that I say. But I will posting a recorded lecture will be I'll finishing up chapter 17, which is blood, which means after Monday's lecture, the concept check of this chapter will also open up. So you can start working on that. This particular chapter's quiz is already open, so you can actually start working on that. And then I also created an extra credit assignment for coming from these two, which will actually give you one extra credit in your exam 1, because exam 1 is not very far away. And this is under exam 1 assignment, exam 1 practice activity. So here is the document. There are a couple of small questions from chapter 16 and 17. This is due January 29. Whenever you are done, you can upload it from here. You can actually get started on this like right after this lecture. All right. And I'm going to post another announcement very soon after I finish this lecture, which will again sum up couple of this stuffs. So don't miss the lecture on Monday. See my recorded video, and I'm going to post this version as well. All right. Any questions that you guys might have before we end the lecture? Any questions? Any concerns, questions? Because personally, I will see you again on Wednesday. We're not meeting on Monday, but you will see the recorded lecture, right? But I will be very much available in email, so you can email me. And I will be back later Monday. I should be back in campus. No questions on the content or anything else? Just watch out then for my announcement and all the videos. I will be posting the next part, which is a Monday's lecture also very soon, right? Maybe by tomorrow or by Sunday, I'll be posting that part too. All right, see you then. Stay safe and stay warm. I will see you then next week. Let me know if you have questions. You can email me anytime. Okay, bye.